The key to this is that there's only 20,000 genes. That's not a large number. And the world is very varied, but they work with the same currency of 20,000 genes, pretty much. That's a revolution.
Today, I'm chatting with Gary Ruvkun, winner of the 2024 Nobel Prize in Physiology or Medicine. As a professor of genetics at Harvard Medical School and investigator at Massachusetts General Hospital, Ruvkun's discovery of microRNAs rewrote the rules of gene regulation and revealed how tiny strands of RNA act as master switches that turn genes on or off. His lab's work did more than just upend textbooks. It opened up an era of RNA-based therapies, from next-gen antivirals to precision cancer drugs, and even illuminated molecular pathways that link insulin signaling to human longevity. So what happens when we can dial gene expression like a thermostat? How close are we to treating age-related disease at its genetic source? And which startups will turn Nobel-winning insight into trillion-dollar industries? This is Ben Kaplan with Deep Venture Partners. As always, our email is deepshow@deepvp.com. Let's go deep. Gary, it's a pleasure to chat with you. How would you describe what microRNA is and why it's proven to be so significant?
MicroRNAs emerged from our genetic exploration of sort of developmental control. How does an animal go from one cell in the little animal we're working on? It's a 1 millimeter nematode. It has 959 cells. There's very few organisms that have that kind of countable number of cells. And so that attracted a lot of people to study the genetics of how does it direct those 959 cells to become a moving, behaving, has a little tiny brain of, 359 cells. And how does that happen? And we happened to bump into these tiny genes that were about 20% the size of any previously known gene. And that was a quirk of a worm for many years, and then we found it was present in humans.
It sounds like you work on these sort of very simple organisms, but then really the floodgates open when we realize this isn't isolated to a worm. So take us through that process, and was that the holy grail moment? I mean, were you searching for this?
You have to think about this in terms of finding sort of unified theories of biology, and the big breakthrough in biology happens when genes can be isolated, and that happened when Victor Ambros and I won the Nobel Prize with Victor Ambros, and we were collaborators. So when we were postdocs, you could pull out one gene at a time and see if its sequence of letters, so there's 20 letters in the genetic alphabet, and you could see if its sequence of letters looks like any genes that were known, but there was very few genes known at that point. There was just a handful. And then, and so we discovered these microRNAs in 1992, but genome projects where all the genes of an organism get identified, those were big operations that involved, 100 people working to generate 3 billion letters of a human genome or 100 million letters of a worm genome, and that identifies 20,000 genes. And basically you and I are something like 20,000 genes, as is a bird, as is a turtle, as is a dog, a fish. Everybody has about 20,000 genes. And it's not that all the organisms on Earth have 20,000 different genes. Most of the genes we have are common to the entire tree of life. Half the genes we have, a bird has or a fish has. And so when we pulled out this little tiny worm gene, eventually, it took about 6 years, but eventually we could ask the question just by sending it over a modem, actually, from home. This was early days of the web, right? 1997, 98. The web is barely going. And 9600 baud sounds fast at that time. It's not slow. It was fast. And so we could send our sequence of a gene, which was, 25 letters long, and ask, is it in the human? And there it was. And in fact, the reason I did it was I was writing a grant application to the National Institutes of Health, the same organization that's canceling all these grants at this very moment, right? And what do they want to see? They want to know that it's relevant to humans, right? If we're working on a nematode, half the people who are reviewing our grant are rolling their eyes saying, well, who cares? It's a nematode. What about humans? And so over a 9600 baud modem, while I'm writing the grant, I went, oh, wow, look at that. There's a homolog. And so, I type away, look, there's a human homolog. And of course that and then the paper, we published the paper 6 months later, and that was a big hit because, seeing that this microRNA was something that goes all the way, it's almost 1 billion years of evolution.
Once that happened and like sort of this process of scientific discovery, how does that, lead to kind of spark really a revolution in RNA medicine that comes later? What are the parts? Is it just more researchers then are looking for more homologs? There's more work done. It attracts attention because this has now broad-based applications. What sort of goes from that moment in time to like the broader RNA revolution?
I think the main thing is it was a sort of a mindset of, oh gosh, genes can be really tiny. And so there were tiny genes before that, or small genes, the transfer RNAs that were so discovered in mid 60s. And these are the RNA genes that are the basis of the genetic code, right? There's a code that takes DNA sequences with the 4 bases of DNA or RNA, and then translates that into protein. That's done 3 bases at a time, and a tRNA does that. It decodes. It's basically a computer program, and those are about 100 units long. And so our microRNAs come from a precursor that's 70 units long, and the mature microRNA is 25 units long. And it just told researchers, you have to think about very tiny genes. And these tiny genes are present in the tree of life, basically starting at the beginning of eukaryotes, that is animals with nuclei, which are the plants and animals and fungi.
And how does then, like the fact that both of you discovered this, I mean, you're a geneticist. There's evolutionary biology components here. What was it about your work and sort of this part of discovery that allowed you to kind of combine things in a novel way and sort of, unveil this novel dimension of gene regulation? What was it about the process, your backgrounds, your approach that made it, discoverable for you at this moment in time?
Well, we were really highly influenced by a sort of an enterprise that was called developmental genetics. So we were studying how is it that genes tell an organism the complex choreography of cell divisions and cell differentiations to make a complex organism like a brain or a muscle or a digestive tract, or in our case, we were dealing with molting of a worm. How does it have a program to, get rid of its cuticles so it can get bigger every 12 hours, that sort of thing. And so we were influenced by maybe 1,000 scientists who were thinking about how does a fruit fly, how does it know where to put a wing? How does it know where to put a leg? How does the salamander know these same things? And this is 100 years of tradition. We were doing it in a worm, and we would talk to each other. And so that was a sort of one cohort. But then when we discovered these tiny RNAs, we got sort of thrust into a very sophisticated community of people who've been studying RNA beforehand. And RNA had quite a great tradition because the ribosome was discovered in the mid-50s. That's the thing that translates proteins out of the code from DNA. And so the ribosome was a huge mystery. What is the genetic code? This is I was born the year of the double helix, right? So, in what we're talking about, when I start studying this, I'm in my 20s. And I didn't realize till recently that this knowledge of the genetic code was only 5 years old when I was a graduate student. I thought of it, hey, it's in textbooks. Everybody knows this, but it was new.
Gary Ruvkun and his co-Nobel Prize winner, Victor Ambros, both worked in the lab of another Nobel Prize winner, Robert Horvitz, at MIT. So do great scientists and great discoveries gravitate to each other? Do birds of a feather flock together? At Deep Venture Partners, we believe that this phenomenon occurs not just at MIT or Stanford or Harvard, but also in hotbeds of deep technology activity around the globe. From overlooked battery tech at Northwestern to materials science at the University of Oregon, and from autonomous aircraft research at Mississippi State to forgotten plasma physics at the University of Strathclyde, great scientific research in related fields congregates together. That's why we spend inordinate effort tracking innovation at more than 1,000 research universities worldwide. And we're not limited by geographic borders either. Why does recognized scientific discovery, why does it kind of cluster together? What role did you play? I mean, is there an example that you learned from working in the laboratory of someone who, of course, goes on to great success as well? Why does it seem to work like that?
Well, it's really cultural transmission. So I was a grad student in the Harvard Biolabs, which is in Cambridge, and that's where Watson went after the double helix. And he was still is a force of nature, and a very opinionated man, very, just awesomely brilliant. And he attracted a lot of brilliant people working with him. I mean, you can't have a mythic status more than discovering the double helix, right? It's kind of the most important discovery in the history of humanity. So,
okay. Okay.
It's like number one, right? And so, and mind you, I didn't know him at that point. In fact, he would already migrated to Cold Spring Harbor, which is a research institute. And keep in mind that the discoverer of the double helix, who made established this department of great people at Harvard, he went on to manage the Human Genome Project. You can't, what's more mythic than that, to discover DNA and then manage the determination of 3 billion bases of DNA? I mean, that's heroic. It's doubly heroic in one lifetime. He didn't have to be reincarnated to do this. He did it in one lifetime.
Okay, sure.
He's still alive, by the way. I'm hoping to see him next month. He's he's a hero squared.
So these kind of figures, attract other people, which creates this kind of cluster of
greatness.
Yes.
And that is cultural transmission. So I'm not genetically related. Well, Bob Horvitz I might be genetically related to, but Victor's not genetically related. We discovered that Bob is from a part of Bielorussia where my grandparents are from. So we actually might be cousins. But I attribute it all to cultural transmission from Bob. He taught us a lot, as did the people in his lab. And he was a grad student with Jim Watson. So, there's this transmission of culture, and that's what universities are. Universities have nothing to do with genetics. It's all about your teachers teaching you. You're not related to them. Right?
What impressed upon you in his lab, and since then, as you've had your own lab and your own postdoc fellows, and you continue to, I guess, either you could just say disseminate the culture or pay forward the culture. What impressed you that you brought from that experience?
There's just this level of commitment to discovery, and a kind of a cadence of continued discovery. And so, down the hall, there was Jack Strominger, who figured out how penicillin worked. And then after 20 years of that, he decided to do immunology. Complete change. And then made important strides in that. And, just an example of ambition, effectiveness. And it's not like running a team of 1,000. These are working with a group of 15, 20 people.
And do you think, there tends to be, and I'll put, because I'm a venture capitalist and I did my, I went to undergrad at Harvard, there tends to be this focus of like, it is these, famous preeminent institutions, you're at one of them where all of this great discovery happens and people tend to focus on that. But do you think that's right? Or do you think, because obviously you're a case of, hey, there's a cluster of greatness and it comes from there, but it's also this, this global ecosystem and work, and there's insights and expertise everywhere. So do you feel that reputation of a Harvard or an MIT or someplace like that is justified, and of course we should focus on it? Or do you think that it maybe gets so much attention that we don't pay attention to maybe other places where these clusters can form?
Of course, there's names like that, but there are other clusters of greatness. And in fact, University of Indiana, University of Illinois, these were the land grant colleges that Congress, decided to fund agricultural research in 1880. It might be 1870, but something, late 19th century. And these became great universities, first specializing in agronomic science. And they're the center of the Green Revolution. But they also, when a whole bunch of great scientists left Europe during the Nazi era, they ended up going to the Midwest. Tons of them went to the Midwest. And their fantastic genetics came out of Indiana, Indiana State, Illinois State, Carbondale. These are fantastic places. And they are just as good, just as important in the history of science.
Do you consider everything that's transpired since? There's been a variety of applications. I think one application that people pay a lot of attention to is, an array of human diseases that this impacts. Coronary heart disease is one that's been talked about, where this plays an important role. What phase are we in now in terms of the science? Is it more this application phase? Is it, does the Nobel Prize in 2024 for you create a resurgence of interest in the area? Where are we headed now?
It's always hard to know where the future is going. But I can tell you that there were some waves of interest. The small RNA revolution, really, the tiny RNA revolution, which is really microRNAs and what are called siRNAs. And these are the related tiny RNAs that do antiviral defense in a plant, for example, or in a nematode. They were discovered about the same time, and it was clear that they used a lot of the same mechanism to make these tiny RNAs. And it's the way plants resist viruses, and it's the way a number of animals resist viruses, not humans. Humans, for some stupid reason, got rid of this system, as did most of the animal kingdom. It's a rare animal that have the tiny RNAs of RNA defense. But it's for sure present in plants and fungi, and it's very important for their defense. And it will probably empower the siRNAs that are used in the biotech industry for treating many diseases, is if you can sort of make the system work better. I think that's where it's going to go.
Meaning that if there was another, COVID pandemic type event, maybe there would be more resistance to that in the future if we understand how these tiny RNAs function better.
Yes, and they are used in a variety of animals, but for sure they're used in plants. I view this through the lens of the genomics revolution, and what that did was teach us that, if you're working on a fungus and how does it decide how to bud or how to divide, you're going to come up with genes that mediate that are the same genes as cause a human disease. And so whenever we make a discovery, like one of the genes we discovered, it came out of our genetics. And the way this works is you do a screen for a mutant that's way different than normal. And it either survives when everything else would die, or it does better. And so that's what genetics does. It looks for variants, which, by the way, is how crops were domesticated, by looking for variants of wild species. Humans were geneticists long before they had factories or, they made metal. The oldest science is genetics. And so when we discover a gene, we determine its sequence and we instantly find its cousins, for example, in a yeast. And that means I might find 50 papers from yeast genetics that have worked on my gene, but they've only looked at it through the lens of a yeast, and I'm looking at it in a fresh way in this little animal. And so I'm able to distill the knowledge from this fungal work or from a plant. And it works back and forth. And the key to this is that there's only 20,000 genes. That's not a large number, right? That is the number of words or number of sentences in the world. And, and the world is very varied. There's lots of different organisms, but they work with the same currency of 20,000 genes, pretty much. That's a revolution.
And is that why fields like, I don't know, like kind of, in a lot of sciences, AI and machine learning are accelerating a lot, but in this particular science, maybe less so because in your words, the vocabulary is more limited, which is some of the great power of all this. Is that the reason?
Well, I don't know. I mean, the AI won the Nobel Prize this year, and it was fun because, we were in the same buses with the AI people. And, they were the stars of the show for sure. And there's a reason for it. And because the using AI, and we got to meet the teams that did it. Demis Hassabis, they created what's called the AlphaFold, which takes protein sequences and predicts how they will fold in 3 dimensions from the known database of 100,000 proteins. And it worked so well, that they got a Nobel Prize like 2 years after they published their first paper about it.
I see, sure.
That doesn't happen, right?
Right.
So that's a serious discovery. Our Nobel Prize was 30 years later. In our case, because our discovery involved RNA, we got thrust into a community of scientists who study RNA, and it happens to be a very Catholic community. And I use Catholic as a term meaning broad. And they for example, whenever you see the tree of life that is constantly shown, the phylogenetic tree of bacteria being the most distant, and all the animals are close relatives, and plants are a little more distant, and fungi a little more distant. That tree is constructed using a ribosomal RNA. That's the most conserved sequence on Earth. And so because it's conserved, you can make a tree out of it, looking, well, who's more different than whom? So the gorilla looks more like a human than a monkey, but a fungus is further and a plant is further. So that community was thinking about the unity of life from the beginning. And I even used that to propose looking for this life on Mars by using that tree, right? I don't think the tree of life that we have on Earth started here at all. I think it came from elsewhere. I think if you go to anywhere in the Milky Way, the organisms are going to fit on the tree of life that we have here.
Imagine every gene in your DNA as an app on the world's most complex operating system. For decades, we thought only big protein code mattered. But microRNAs are the tiny scripts that hot-fix those apps in real time. Need to throttle runaway cancer? MicroRNA can block the tumor. Want to reboot metabolism? MicroRNA can reduce insulin signals after a big meal. Fighting a virus? Custom microRNAs can shred viral RNA like digital malware protection. Using this research, biotech firms are now editing cellular behavior without touching the underlying DNA, promising therapies that are faster to design, easier to reverse, and cheaper to manufacture. And you've referred to it as like a computer program, even in this conversation. Why do you view this like a sort of, I don't know, the tree of life as a computer program? Why is that a good analogy for how things operate?
Well, I mean, DNA is a script. You know, to make a bacteria, you need 4 million bits of code, and to make a human, 3 billion. So it's about a factor of 1,000 more complicated. And so some people say, well, gee, bacteria are so simple. Who cares? They are not simple. Bacteria are just the most unbelievable organisms. They're so great.
In terms of microRNAs, you know, and therapeutics, which is something that we pay attention to, like what is the direction that they need to go? Like I'm talking microRNA therapeutics before, you know, they might displace or outcompete other therapies. What has to happen from the side of sort of bringing them to market, commercialization? Where do we have to get to with microRNAs?
Well, I think we need to have more genetics sort of saying there are mutations in microRNAs that cause diseases. And so, you know, microRNAs do important things for sure in plants. They're like the domestication of corn is a mutation in a microRNA that leads to a big ear of corn. That's kind of important in human history. And that is a microRNA mutation that led to going from teosinte to corn that we love so much. And so it tells you that at least for that in plants, it's been really important. But that hasn't happened so much in human genetics. It's been a little bit with coronary heart disease, but it hasn't yet sort of been an explosive finding. And that part of that is that the way a microRNA base pairs to its target, so it uses, you know, Watson-Crick base pairing, but it doesn't use all the bits. It's 22 bits long, but it has bulges and loops, and so you lose a bunch of bits, which makes prediction of targets hard. And so that's been a sort of slowing up what's being done in humans for finding genetic disorders, for example. Whereas in plants, the microRNAs find their targets with perfect duplexes. And so finding their targets has been a real gold mine.
The application side, the medicine side, the commercialization side, who is suited for that? When you sort of observe others, and I don't know, maybe like there's a grad student who goes on and tries to commercialize something, or there's, you know, other business people with experience in science who do this. What, you know, do you have a perspective of what works on that side? I know you don't do that directly, but what do you observe?
I don't not do it because I'm against it. I just, my expertise is just in a different dimension. But, you know, like Wally Gilbert's one of my buddies, and you know, he did pure science, you know, figuring out how gene regulation works in a bacteria, and then went on to start Biogen in Boston, which is a big deal, right? It's a big company. He and Phil Sharp were sort of the early biotech guys. Both of them are, you know, Nobel laureates, and were part of the recombinant DNA revolution. You know, Wally invented DNA sequencing, and Phil Sharp discovered splicing, and you know that eukaryotic genes have introns in them, and they get spliced out. Big, important fundamental discoveries. Unclear where they were going to go in biotech. And then Phil Sharp, at age 55 or something, joins the tiny RNA revolution. He starts coming to all the meetings where we're talking about microRNAs and siRNAs because he thinks it's important. And he starts a company called Alnylam, which is a tiny RNA company for making drugs, because he thinks it's going to be the next revolution. And so he's super entrepreneurial.
Who is suited for like the pure science and who is suited for like a commercial application?
When people come to my lab, they think that I'm going to say, oh, you have to go academic. You have to be a professor. If you go into biotech, you're wasting they think I'm going to be like that. And I'm completely agnostic. To the people in my lab who've gone to biotech love it. None of them say, oh, I regret that decision. Not one. And there's a third of them have done that. And so the people, I have postdocs now, and I say, yeah, if you're thinking about your career, biotech is completely fine. And they think I'm going to be, oh, no, do a university track. No, that's like one of three choices, right? You can go pharma, you can go teach at a junior college, you can teach at a state university, or you can go start companies or work in pharma. And I tell people that if you go to pharma, you probably spend more time on science than if you have an academic lab, because I spend a third of my time raising money. If you're in biotech, somebody else is raising money. You don't have to raise the money. You're doing science.
Well, speaking of which, recent developments, funding available for scientific research. Obviously, you're someone who, has, as you said, leveraged, made a career in this. Are you concerned about current cuts?
I turned in my renewal of a grant, which studies frame shifting of proteins in how a worm knows that it's being attacked by a bacteria. So it's all about infectious disease and how do you start to mount a response to a bacterial infection? That's kind of relevant. And it's good science for sure. I mean, I'm not objective. It's my science. So maybe, who knows? Maybe I'm misguided. I don't think so. So I send in my renewal. And it should have been renewed. But of course, it hasn't been reviewed because NIH is shut down. And I just sent in a renewal of my tiny RNA grant. It's been going 32 years. And okay, some of your listeners will say, well, geez, you've been on the dole. You're a welfare case for 32 years. Get a job. But I could also tell people, your listeners, that my grants are $300,000 a year, and it supports 5 people making $60,000 a year who have PhDs. That's what a painter or a carpenter makes, right? So the people in my lab are being paid like sort of manual laborers. And I'm not saying that they should be paid more. I'm just telling that they are not paid a lot.
You're saying this grant, this renewal of this $300,000 grant is a great potential return on investment for the country, humanity, and everything else. And it's pennies. It's pennies on the dollar.
To say it in a Boston accent, it's a baggin'.
Okay. Okay. Haven't heard that in a while since my undergrad days at Harvard, but yes.
If you go over the 30 years I've run a lab, it's always been with students and postdocs, and nobody is making $1 bazillion a year. So, yes, it is a government investment, and you could debate whether government should do that. But the U.S. government decided to support science after the Manhattan Project because, hey, we won the war for you. And what did MIT do during the war? They developed radar. This is kind of important. Scientists do important things, folks. And, we didn't lose World War II. Why do you think that is? There were people on the ground, but there's also scientists solving things. In fact, the guy who discovered the ribosome, who named it, his name is Richard Roberts. He died, and I read his obit. During World War II, he developed the proximity fuse. So before he discovered the ribosome, he discovered a way to send a vacuum tube on an artillery shell so that it would know when it's near metal and explode. And it made artillery shells 10 times more effective than they were in 1943. That's science. That beat the Nazis.
And I think your story and hearing that from you means something, because this idea that the applications of science are not always obvious. Sometimes some of the most important applications start out in a completely different way, but you have to make the commitment to invest in science, and the commitment particularly to those who have already demonstrated that they can do amazing things. And so what I worry about sometimes is that there's a role, and I'm a venture capitalist. So, there's a role for we come in with funds when things are ready to be commercialized, and we want to push it all the way to market, and it's a great opportunity. But to get to that point, a lot of things have to happen first. And how much of that core scientific understanding has been funded by some type of government grant that paved the way, and maybe we were studying a worm at the time, but it has implications eventually to a wide range of diseases that can transform human health as we know it. We have to invest.
Is a success story. I mean, the idea that this is the worst government agency in the U.S., it's just hogwash.
What is your final from this entire journey that you've been on? What is your advice for not only other scientists and academics, but others who want to pursue innovation, others who want to think in novel ways? What is the lessons of this that you take with you, that you share with your science friends, but also some of maybe the non-science people in your life from this, kind of Nobel Prize winning journey you've been on?
That there's magic in the world, and not every science paper you read is magnificent. There's a lot of really average, mediocre stuff, but there's the top 10% of stuff is it knocks your socks off, right? And there's just a lot of high quality work that kind of is obvious to many people, I think. But where do you learn that? It's a kind of connoisseurship. It's like knowing what's good music. It's just experience and hearing a lot of music. It's hearing a lot of science over the years. And I tell people in my lab, what you're going to remember is the 100 seminars that you heard, over the 6 years that you're in my lab. Not, a few will be in our group meetings, but a few will be when a guest speaker comes in and talks about how hybrid corn can be bred from this and that, with this mutation and that mutation. And these are things you remember.
When you kind of embrace the love of science, that's a good analogy. It sounds like music, and you can start to hear the music, and it can change your life. So thank you for joining us on Deep.
Pleasure to talk with you, Ben.
Thank you to Gary Ruvkun, winner of the 2024 Nobel Prize in Physiology or Medicine and professor of genetics at Harvard Medical School. Be sure to subscribe for the latest episodes and message us at deepshow@deepvp.com. This episode has been brought to you by Deep Venture Partners, an emerging venture capital fund in Silicon Valley. Get show notes, transcripts, helpful links, and more at deepvp.com.